Hello, 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 hello. If anyone is there, feel free to say hello. We are going to be looking at some stuff on genetics. So I thought everyone tends to hate all the genetics things. So I thought I'd try and go over all the tricky parts of genetics today. Who's here? I've seen one comment, I think, so far, which is a bit useless. Not that you are useless, Sabiashi, but hey, how are you doing? Hey, Fadia, how are you doing? All right, how was your day? Hey, Benny N, you all right? Does this include DNA profiling? It does not include DNA profiling. So I will do a session on, I don't know, the hard sort of applied genetic things at some point, but not today. Oh, yes. Hello from India again. Hello. Matilde Scrivener. I used to have a teacher who had a very similar name to you, which is interesting. Anyone had a good day today? Anyone been sent home yet because of coronavirus? Epigenetics is not in this topic, no. Hey, right, should we get cracking? Why dilly dally? No one's telling me how they're getting on. Oh, it's you, Tilly. Hey, Tilly. What are you doing? Good question. So let's get cracking, eh? This is mostly year 13 stuff, yeah. I don't think any of this is really year 12. I've also decided to skip out some of the easier genetics bits. So hopefully by the end of, I don't know, in 45 minutes time or in an hour's time, let's say, I would like every single one of you to know what I mean by sex-linked inheritance. And for those of you that are doing, who are like ultimate Snap Provider subscribers, I'm going to be covering this bit here and this bit here a little bit later. Just, I know, possibly not that last one, definitely the first one. So you don't necessarily need to listen to this first bit for those of you, but everyone else, you need to listen. We're going to look at autosomal linked inheritance and we're going to look at epistasis. So both of these two are all of the specs. This one is not covered by EdXL. So if you do EdXL, you can basically just go off when you're done after I finish autosomal inheritance, and then we'll be done. What else are people saying? Can I talk about gene replication? I can, but I won't. I haven't covered any of this. Fair enough. This is really hard. This is probably what I consider to be the hardest part of A-level biology. And catching up, yeah. If you go on YouTube, I think most of them will have like a page that looks something like that on them. If they're one of my little lessons, it will have something that looks like this at the very start. So watch out for that. The last one I did was, I can't even remember. I've done kidneys, photosynthesis, respiration. Can anyone tell me? What have I even done? I can't remember. I don't know. Gone. Anyway, so for those of you that are interested, that was it. Nerve impulses. Thank you, Blair. I totally forgot about that. For those of you that are interested and you want to check, these are the spec points that you want to basically compare to. So after I've gone through this, you want to be checking if you understand things compared to this. So this is the stuff I'm not going to be talking about today because I've sort of deemed it a bit too easy. So if you are really stuck with genetics, I would probably look at this sort of thing first, but I'm not going to bother with that today. So fast AQA, OCR, very, very similar. So I'm going to be looking at this sort of stuff, the harder bits. And then EdXL, similar again. I'm going to be looking at the linkage stuff. Can't even see where it says it. There you go. This sort of stuff down here. And then EdXLB, I'm going to do just everything from EdXLB. Okay, everyone good? Everyone happy to get cracking? Don't care, I'm going to do it anyway. So first thing we're going to look at then are sex-linked genes. Do you know what is covered by WJEC? No, I don't, unfortunately. I've sort of made all of these based on the 4 sort of more commonly done specs. So I'm sure there's some overlap, but I don't actually know, if I'm totally honest. Sorry. Anyway, so sex-linked inheritance is where we're going to start off then. So out of interest, does anyone know what I mean by sex-linked inheritance? Aisha has some idea. Tell me more. Sorry if you are from Wales. I am technically probably about a quarter Welsh, so I feel your plight. Although I am supporting England at the weekend. Only on same chromosome, XXXY, meiosis, linked between sex chromosomes, gender determination, linked on the X chromosome. Okay, some of you are here, some of you are right then. So essentially, sex-linked inheritance is where you find a gene which is on the X chromosome. So I'm sort of assuming quite a lot in terms of your knowledge. So I'm assuming that all of you know that you have 23 pairs of chromosomes. And I'm assuming that you guys know that the 23rd one is the sex chromosome. So either an X or a Y. If you are a male, you are XY. If you're a female, you are XX. So when does it occur? Let's define this as saying it is when a gene is found on the X chromosome. Okay, so it's when a gene is found on the X chromosome. That's pretty much as simple as it gets to. I think it is important to note that because men are XY and women are XX, essentially, if men have one copy of a sort of dodgy allele, so the not quite functioning allele, then essentially men will have the disease. Okay, so because men only have one chromosome which contains it, because it's the X, not the Y, then if men have a faulty allele, then they will have the disease, whereas women need to have two to have the disease. So women are less likely to suffer from these diseases. Okay, what are we saying? What are we saying? Will this include Punnett squares? Yes. Good. So someone has mentioned here then that this is because the Y chromosome is a little bit shorter. So there is a bit of Y chromosome, so the bit which would be here, which is not there. So I'll rub it out. That bit is not, it doesn't have like a homologous partner, so that's why there is a problem. So an example of this is haemophilia, which is a really interesting disease, and it's one which used to be quite prevalent in the royal family. Essentially, Queen Victoria was a carrier of it, and lots of her offspring therefore ended up sort of getting it. But essentially, haemophilia is where your blood doesn't clot properly. So if you have a cut on your skin, you could literally bleed to death because your blood won't clot properly. If it's internal, it can be really dodgy, really dangerous. So in terms of what the different alleles are then, X and then like a superscript H, this means that you have a functioning gene, you have a functioning allele, okay? So XH equals healthy. X little h equals you have the disease, so haemophilia. And then if you are Y, there is just no allele here. So if you are a Y, this is sort of a total lack of the gene. Now, it's also worth saying that haemophilia is a recessive disorder. So you need to have two of these faulty alleles for this to be a problem. Okay, how are we getting on so far? Is this okay so far? This is nothing like Down syndrome. It's totally different to this. What else can we say? Healthy equals carrier. Healthy doesn't necessarily mean carrier. Carrier is where you're heterozygous. You have both alleles. Okay, everyone seems fairly good so far. So if I had the genotype, X big H, X little h, or X big H, X little h, what would my phenotype be? Just nice and simply. What would my phenotype be if it was X big H, X little h? Or X big H, X big H? That is dominant, yeah, but what's the phenotype? My face is covering it. Oh no, right, apparently you can't see up here. If my face is covering it, I can only apologize. And I can't really move my face very easy. I think you guys might be able to move my face. Oh, probably not. Oh, well. So if it's X big H, the phenotype is a healthy female. I like that. Well done for mentioning the gender as well. So this is a healthy female. If it's X big H, X little h, then that is considered to be a carrier. If the genotype is X little h, X little h, can you tell me what they would be? So X little h, X little h. Yeah, piano person, able to clot blood is good. Aisha, I don't understand what you're talking about in terms of switching of the X chromosome and all that. X little h, X little h, yep, and gender as well, please. So this will be a haemophiliac female. Well, I just told you. Whoops. So haemophiliac female. X big HY. Well done, Melv. Okay. Sorry, I was just reading. My brain was sort of just functioning there. So X big HY is going to be a healthy male. And then finally, if I've got X little HY, what am I talking about? Good. So I'm talking about a haemophiliac male. Okay, now that's pretty much as easy as this gets. It does get a little more complicated when we start looking at these sort of chains and when we start looking at these pedigree charts, which is what they're called. So looking at this then, I've got a family, which you can see, and you're expected to be able to tell me what the evidence is that this is sex-linked. So I guess before we do that, let's draw the genetic cross between A and B. So if I've got A and B, what are my two genotypes for A and B? Sorry, let's just check where we can do this. So what is A, what is B? Nothing is on the Y chromosome. So A is a, are you sure? Good. So if it's affected, it's X little h, Y. I know you can't do the superscript. And then B is going to be X big H, X big H. Okay. Are we cool with working out what the gametes are? How do I work out what my gametes are? Does anyone know? Where's B coming from? So good, Ghosted Guna, you are totally right. In terms of working out my gametes, all you're doing is separating these. So the male gametes are going to be either X little h or Y. So X little h and then Y. And then my male gametes, sorry, my female gametes are going to be just separate them, X big H and then X big H, right? They are my gametes. So in terms of doing my genetic cross, if I were to do it properly, X big H, X little h, X big H, X little h. The convention is just to put the capital letter first. There's no other reason why I'm doing it like that. X big H, Y, and then X big H, Y. Okay, so looking at this cross then, what is, or looking at what I would expect, what is the evidence here that this is sex-linked inheritance? How can I tell without knowing, without someone telling me that this is sex-linked? Does anyone know this? This is quite difficult. And this is a pretty standard exam question. Disease phenotype, haemophilia. Do you know that bit when meiosis or mitosis is going on and one X chromosome is switched off because a female has two? Oh, right. Yeah. No, I'm not going to talk about that. Which isn't that helpful. Female's carrier, all on the X chromosome, X and Y are involved, XY chromosome. No females have it. Okay, no females do have it here. That's not evidence of it being sex-linked because a female can have it, right? A female could be XH, X little h, X little h. So there is some evidence here that this is sex-linked. I don't know if you guys know what this is, though. So I'm about to tell you something which is going to make your life much easier if you ever see this exam question. Nope. Okay, so evidence then. If we have a look at this, if the male has it, so if the male has it, what do his daughters always look like? If the male has it, what do the daughters always look like? Male give it to their only child. Good. So evidence of it being sex-linked. So what you are looking for, whenever you are asked the evidence, you are looking to see what the daughters of the male person is like. If a male person has it, then his daughter has to be a carrier. Okay, so daughters of affected males equal carriers. So they will either be carriers or they will have it. So have a look at the Punnett square. Look, because if you are female, you need to have two X's. A female cannot have this Y, so they have to inherit this X from their dad. So they've got to have an X little h, and then depending on the mother, they will either have it or they'll be carriers. In this case, they are carriers. If I changed this to a little h, then it would mean that this would be a little h too, and you'd end up with a female with it, right? So one bit of evidence that this is sex-linked, and a really good tip, is that the daughters of the father have to be carriers if the father has it, okay? The other thing that we can look at is we need to look at this couple here. So what can we tell about this couple here? If the mother is a carrier or has it, what will her sons look like? So if a male was unaffected, what would the evidence for sex linkage be? It would be hard to work it out in that sense. Evidence, if the father has it, then the daughter has to be a carrier or they'll have disease. Why the daughter of the father and not the son? I'll get to that in a second. They'll be affected. Good. So if the mother, so here is the mother, this circle, if she is a carrier, so she is X little h, X big H in this case, if she is crossed with a normal man, so a healthy man, then if we look at their offspring, then there is a very good chance that their child will have it. So X big H, X little h. In fact, I don't need to look at the X's. I'll do them very quickly. X big H, X big H. But looking at the the sons, X big HY and X little hy and X big HY. So if the mother is a carrier, then there's quite a high chance that her son will have it. Okay, so some other evidence, sons of female sufferers, don't know if that's the right word to use, but I'm going to use it. So sons of female sufferers / carriers are likely to get it. In fact, that's a bit of a stupid sentence. If it is the son of a female who has the disease, the son will definitely have it. So if your mum were colorblind, then if I'm talking to a boy, then you will have the disease, right? So that'd be a good way of working out paternity really quickly. If the mother is a carrier, then the son is quite likely to get it. Okay, only 50% though, yeah. So in terms of males, if your mum is a carrier, there's a 50% chance that you'll have it. If your mum were not a carrier and your mum were to have the disease, so it was X little h, then you would be certain to have it, right? You couldn't be a son and not have it. Okay, so these are the sort of really important things to take from this. So the evidence of sex-linked disorders, and what you need to remember for your exams is if the father has it, so if the father has it, his daughters will be carriers. And if the mother has it, so let's make this like fully, pretend that's fully black. If the mother has it, then her son will definitely have to have it too. Okay, so that's how this basically works. If you are colorblind as a male, then your mother has to be a carrier or she has to be a sufferer, right? So if you are colorblind as a male, it's come from your mum, because you can't inherit, looking at this, right? You can't inherit the gene from your dad because you only get the Y chromosome from your dad. So therefore it must come from your mum. Okay, how are you guys feeling with this? I know it's complicated. Yes, Blair. Feel silly. Okay, some of you find it hard, some of you finding it straightforward. I'm seeing more ones than twos. Is that why males are more likely to be colorblind? No. Males are more likely to be colorblind because they can't be carriers. They either have it or they don't. That's it. Okay, cool. Right. I've got a very hard question for you to have a go at. Prepared to basically forget everything I've pretty much just told you as well. So in birds, males are XX. Confusing. Females are XY. So bear that in mind as we go through this. Use this information to explain why recessive sex-linked characteristics are more common in female birds than male birds. So why is it more likely that a female bird will have a sex-linked characteristic? If a father is a carrier, the father can't be a carrier, Fadia. I'm not going to talk about Y-linked, no. So why is it that it's more common in female birds? So only one X chromosome. Okay, so that is true, but it's not the full answer. Females have one X chromosome. So what? That's what your examiner will say if you write that. Or they won't. They'll just sit there and say no. So they only have one allele of the X. Okay, so I like that. Females only have one X chromosome. So one allele on the X. True. So what? If they have a faulty allele, there is no backup. Missy, that's really, really good. You want to word that a little differently for A level. So that's what you could get away with at GCSE. For A level, you want to say only one recessive allele needed to be expressed in the phenotype. Okay, so because they only have one X chromosome, the females in this case, then they only have one allele and they only need one recessive allele for that to be expressed, right? If you were a male in this case, you need to have two recessive alleles, whereas the females will just need one. Okay, so you're basically a bit screwed. Yeah, this is why mammals, in terms of females, are genetically superior to males because they're less likely to get certain diseases. No, there's no way around that. Okay, so that is how you'd have to write this. If you just said females have one X chromosome, you will not get that mark. You need to say that they therefore only need one recessive allele for it to be expressed in the phenotype. Okay, so in chickens, a gene on the X chromosome controls the rate of feather production. The allele for slow feather production, F, is dominant to the allele for rapid. So F equals slow, F equals rapid. Okay, the following figure shows results produced from crosses carried out by a farmer. So he's got a male, which is slow feather production, and female slow feather production. And they've had some offspring which are a bit mixed. Okay, so explain one piece of evidence from the figure which shows that the allele for rapid feather production is recessive. Yeah, this is year 13. How do I know this is recessive? Only females have it. That doesn't tell me anything. As it is expressed in parent-free. It's expressed in parent-free, so it must be a carrier. Parent-free is not. Parent-free is a complete carrier. They just have it. No males are affected. Guys, I'm not asking you why it's sex-linked. I'm asking you why it's recessive. Yeah, this isn't asking about it being sex-linked. You're looking for an answer, which I told you earlier. This is a totally different question. This is hard. It is very hard. How common it is? Yeah, I see your point. You'd get that at GCSE. You wouldn't get it at A-level, though. 1 and 2 are not affected. Jasmine, tell me more. You're very close. 1 and 2 are not affected. However, children 9 and 10. What we're looking at with 9 and 10. Children 9 and 10. Oh, I see. Yeah, 9 and 10 don't have it, but 3 does. Yeah, that's some pretty good evidence. 1 and 2 produce kid 5 with affected, which means 1 and 2 are carriers. Lovely. Really, really good. Yeah. Well done, guys. So explain one piece of evidence. This is a good enough piece of evidence. 2 people, or 2 chickens in this case, 2 chickens that are both normal phenotype, they're both slow feathered, end up with an offspring, number 5, which have the thing, they have rapid production. So in terms of wording this, you'd want to choose your chickens, right? So you'd want to say chicken 1 and 2 are slow feathered producers or something. So 1 and 2 are slow feather producers, but you want to say chicken 5 is fast. And then to get your second mark, you need to say that this means that chicken 1 and 2 are what? So they are carriers. What's another word we can use for that? Heterozygous. Yeah, lovely. So 1 and 2 must be heterozygous and pass on recessive allele. Okay, so they're heterozygous and they've clearly passed on their recessive allele, because otherwise their son wouldn't have got it. Okay, so that's something you could say. I'm trying to see if there's any other marks I could possibly give. So that's the one that they're more keen on you talking about. I guess if you could argue the case properly for 3 and 4 and say that their offspring haven't, so 3 must have be is recessive, so it's passed on its allele, and 4 basically, no, it's harder to do. 3, you could argue, you'd have to talk about dominant traits and how 9 and 10 would have to have it because they've got a dominant trait from their mother. But 1 and 2 are, I don't know what they wanted to say in the mark scheme. No, ratios don't help you in this question. So give all the possible genotype. Oh, no, yeah, you could get ratios, sorry, if you mentioned this. So if it was dominant, then you'd expect 9 and 10 to also have it, right? You'd expect that to be the ratio. It would be just 1. But because they don't, it shows it's got to be recessive. Give all the possible genotypes of the following chickens from figure 1. So what is chicken 5 going to be? Well done for remembering that it's X and Y for chicken 5. In fact, so chicken 5 is female rapid feather. So it means it is X little f Y. I agree. Yeah, so little f is for slow and 5 is sorry, little f is for rapid, and this is rapid. Chicken 7, this one, what do we reckon? X big F, yeah, good, well done. So there are two options here. We don't know enough. It's either X big F, or it is X big F, X little f, right? Because we don't know what their offspring are, there are two options, right? Or we do know what the offspring are, but we don't know if these lot are carriers as well. So 7 could be one of two things. So to get the marks, you'd need to say both, okay? So a cross between two chickens produced 4 offspring. 2 of these were males with rapid feather production, and 2 were females with slow feather production. Give the phenotypes of the parents. Chicken 5 is female, I agree. Remember that it says here that females are XY. You can't tell from 7's parents. You can guess, but you can't tell. There's like a few ratios. How are we getting on? So question number 3, yeah? So 2 offspring are females with slow feather production, and 2 are males with rapid feather production. So what are the genotypes of the parents? I'll show you how to do this question. So to do this question, you want to write down what the phenotypes are of the genotypes of the offspring, and use that to work out the parents. So 2 are males with rapid feather production. Okay, so I want to have an X little, wait, where I've lost it. So it's X and X, and rapid is too small. So X and X little f. And then my females are going to be slow and female. So XY, slow is big F, isn't it? Yep. Right. So in terms of my parents, then, I know that, and I'm not going to write any more than this down just yet. I could write a bit more than this down, but for now, I'm just going to leave it like this. So in terms of working out the parents, I know that one of the parents has to be X little f and Y. Right? That's got to be one of the parents, because you got to get the Y from somewhere. And then for the other one, I need there to be, in fact, does that make sense? Don't think that does make sense. Where have I gone wrong? Oh, that's it. Okay, so one of them has to be X little FY, because you can't have basically this combination any other way. Let's put this one down here for now. So X big F Y. So in terms of my other one, this has to be this has to be a little f here, and this one has to be a big F here, because that's the only way you can make these combinations. So that means that this one is big X big FY. So that's one that I need. So I need a Y from here, and I need a big F from here. And for this one, my X little f, it needs to look like this. This means that there are some other combinations I can get too. So it means that this one would be X big F, X little f. And it means that this one would be X little f, Y. So in terms of this, it says there were 2 females with slow feather production and 2 males. It's not saying that the ratio has to be 1 to 1. It's just saying that 2 of them were made, and that's got to be a possibility. Okay. So for my answer, this is really confusing, this question. I totally agree. And I don't doubt that it's me that's somewhat confusing you in this process. To get this answer, in fact, have I just messed this up? There's a good chance I've just messed it up. Oh, no, I haven't. So in terms of my answer, it's got to be X little f Y as the male, sorry, the female, and it's got to be X big F, X little f. Okay, so the way you've got to do this is you've got to look at the genotypes. So male with rapid feather production. So rapid is little f's and male is XX. So there's one of them. Right, so I've written that down, and I can't have a big F next to it here for my female, because I've only got a little f, so that means it's got to be below here. So my X big F, because I've got a female with slow feather production, slow is big F, so it's got to be X big FY, so it's got to be down here. I then just need to put the other bits together. So I know these two, which means I know what this phenotype's going to, I know what this, allele is, and then I know what this allele is going to be. And I also can work out that one, and you just need to put the other ones in. So what are you guys saying? Why have they reversed chromosomes to male and females in the question? What are they trying to do? This is just the case for chickens and birds. Birds just basically are the other way around. You've seen this practice question before. Yeah, it's a hard one. Confusing because the other way around to mammals. I know it is very confusing. Could you over the last one from the start? I would say have a go at watching this again on YouTube because otherwise we won't have time to finish. Okay, so have a go at watching this again at the end because otherwise I just won't be able to finish. And I'd rather do a little bit more. So I'll very quickly try and say it because I don't like leaving people confused. Basically, step one, write down the two genotypes of your offspring. As soon as you've done that, you realize that this one can't be next to this one because you couldn't have a little f causing this to be a big F, right? And then you just need to fill the rest in. So just give it a go. And if you get really, really stuck, I might be able to help at some point in the future, but possibly not. It shows, yeah, it does. Yeah, Aisha, you're right. My cross does show a male, which is going to be slow, and it shows a female, which is also going to be fast. It doesn't say that these are the only options. It just says that these are two possible options that came out. Okay, so there was a 1 in 2 chance that you'd get these offspring compared to the others. Anyway, moving on. P-values. I will actually talk about that at some point today. So next thing I want to look at then is autosomal linkage, which is also horrible. So what do you guys know autosomal linkage to mean? I honestly have no idea about MJEC or MJAC or whatever it is, so sorry. P is not the Hardy-Weinberg equation for this. Oh, it is sometimes, but not for this. When 2 genes are on the same chromosome, lovely. So autosomal linkage. Just so you know, an autosome is a type of chromosome that is not a sex chromosome. So 2 genes found on the same chromosome. Here you go. So what have I gone for? F and D. I did come up with something for F and D to be, and I forgot. Oh, there you go. Freckles and dimples. So they're both found on the same chromosome. So here is the maternal one. So these are homologous. Here is a paternal one. So F, we can say that big F leads to freckles and a big D leads to dimples. Little f leads to freckles, and then little d leads to dimples. So what does this mean then? So yeah, there are 22 autosome pairs, correct. What does this mean? If you have 2 genes on the same chromosome, what does it mean? You could say they are the same, they're different loci. Same chromosome, but different loci, because loci is just a position. Both expressed? Not necessarily, but what are the chances? If they're on the same chromosome, they are more likely to be inherited together. Lovely. So these genes, or let's say, yeah, let's go for genes. In fact, I think I prefer the word alleles here. So these alleles are more likely to be inherited together, right? So because they are literally linked on the same chromosome, if this is more likely to be in a gamete, so like a sperm or an egg, it means that if you have freckles, you're also going to be more likely to have dimples because they're on the same one. If they're on the same gamete here, then if you don't have freckles, then it's also more likely that you won't have dimples. And the closer they are together, the more likely they are to be linked. The further away, the less likely. Okay. So if we move on then, if I had 2 pure breeding organisms, that basically means they've been bred together so many times that you can be almost certain that they are homozygous dominant or homozygous recessive. Okay? So pure breeding organisms are bred together. So I've got this guy over here, which is big F, big F. In fact, I'm giving away an answer to him. So looking at the genotypes, what are the genotypes of this one on the left and this one on the right? Cronus, I'm not going to randomly tell you how to do something that I'm not talking about right now. Sorry. Good. So my genotypes are F, F, D, D, that one. And this one is going to be little f, little d, little d. What are my gametes going to be then? You guys are ahead of me already. Well done. Oh, not quite though. What are the gametes? Good. So FD is one gamete here, because normally we do this thing, don't we, where you do this little crossy thing that looks like a parrot's claw or a crab claw or beak or whatever, you know what I mean. Normally you do this. If they're all the same, you only need to do it once. So that is one gamete. This one I've got a little f and a little d, little f, little d, little f, little d, little f, little d. So something like that. Okay, to save you some time, if I had this cross, it's really, really easy. I don't need to draw it out like 16 times either as you're used to. FD and then little f, little d. Here is my cross. All the offspring are big F, big D, little d, right? If I did more of them, like if I did another F, big D here and another little f, little d here, you still get the same thing happening, right? I'm just saving myself some time and not having to bother drawing all of them because they're all the same, right? If the gametes are the same, you don't need to draw them more than once. So they are my gametes. If I were to cross my F1 generation, F1 means the first filial generation, okay? So if this is my genotype now, I've got big F, little f, big D, little d. And I'm going to cross that with big F, little f, big D, little d. Essentially, all this means is one of my chromosomes, because you have two, because you got one from your mum, one from your dad. One is big F, big D, because they're linked. And the other one is little f, little d, right? Same here. So big F, in fact, let's draw all of them. There you go. Okay, so if I were to write down the genotypes, I've just done that. What are the gametes that I produce here? So what are the gametes if I've got big F, big D, little d? Cronus, I like for you're saying you're lucky that you're not doing A2 just yet. It will happen. Well done, guys. Some of you have clocked, some of you haven't. So lots of you are saying that you'd have big F, big D, big F, little d, little f, big D, and little f, big D. However, because they're linked, it means that I can only have big F, big D, or little f, little d, or big F, big D, or little f, little d, apart from crossing over, and I'll get there. So because they're linked, it means that my gametes are actually still the same. So I've got big F, big D, big F, big D, and I've got little f, little d, and I've got big F, big D, and I've got little f, little d. So if I draw the Punnett square then, let's just make this a bit bigger. So they were the offspring, they were my different gametes. Big F, big D, little f, little d. So I've got big F, big D, big D, big F, little f, big D, little d, big F, little f, big D, little d, little f, little d, little d. Okay, so because it's linked, how do you tell if it's linked? You tell if it's linked because it literally says in the question. It will always say in the question that the genes are linked. So there's another way of knowing, apart from it will tell you it's linked, or maybe they'll show you a picture. Okay, but they are linked. So if we have a look at this, essentially what I've got is a ratio of this one has freckles and dimples, that has freckles and dimples, that has freckles and dimples, and that one doesn't. So my ratio here is going to be 3:1 of freckles and dimples to those that aren't freckled and dimpled. That's if we're looking at the phenotypes. If you were asked to look at the genotypes, then it'd be 1 here, 2 of them here, so 1 to 2 to 1, right? So depending on what they ask, and it's normally phenotypes, it's either 3:1 or it's going to be 1 to 2 to 1. Okay, yeah, you do associate this with monohybrid inheritance, but it also happens with autosomal crosses too. Okay, so this is never actually the case. So if we have a look at this diagram, this is again quite difficult, but I'll go through it with you, obviously, because that is literally my job. If we have a look, the expected values that I wanted was going to be 3:1. However, this is almost never the case. Now, the reason it's never the case is because when I'm making gametes, and you guys should remember this from meiosis, we have a process called crossing over that occurs. And essentially what happens is parts of the chromosomes are basically going to kind of intertangle themselves or something. And then you end up swapping a bit of the chromosomes about. So this started off as being little b's, but it's now swapped with one of these big B's, right? So because of this, I started off where you could only have big H and a little b on the same chromosome, right? So I think this is actually a different example. Yeah, this is a different example. So if we go for H and B, we can call them having brown hair for the bee and a hitchhiker's thumb. If your thumb bends backwards like that, it's called a hitchhiker's thumb, right? If these two are now shared, then normally I'd expect that my gametes would either be big H little b or little h big B. However, because of crossing over, I've actually made a gamete here, which is little h big B, which is apparently the same. Have I messed this up somewhere when I've made this? Possibly. And then this one over here is going to be big H little b. Yeah, what have I done here? How have I messed this up so bad? Something has gone wrong, right? Or am I being an idiot? Tell me if I'm being an idiot. Yeah, this is similar to independent segregation, but slightly different. Big H little b, big H little b. Where's that one gone? That one's come down here. Yes, I am. Am I being an idiot? I'm pretty certain I'm being an idiot. That was because this one was all big B's. Oh no, yeah, so little h and a big B, but that was. I'm being an idiot because they are the same, are they? You mixed up the H's. Oh yeah, cool. I've mixed up the H's. Excellent. Thank you for telling me. So yeah, this should be a little h. That one there should be a little h. Thank you for working that out. My brain was definitely not working quick enough to do that. So I've made up something over here, and this one should be a big H then, shouldn't it? Thank you. So essentially what I've ended up doing is I've made some gametes, which are going to be big H, big B, and I've also made one which is going to be little h, little b. Right. So essentially, this autosomal linkage, you should be getting this ratio of 3:1. However, due to crossing over, you can end up basically having something which is a little bit off because crossing over doesn't work out perfectly. Right. So some gametes, and this is what you need to say in an exam question, which I'll show you in a second. Some gametes are made which don't have the linked alleles on, right? And this is because of crossing over. Yes, I am clearly a massive spanner here, and I have managed to mess this up. Sorry about that, guys. I'm just going to ignore me. So, right, how are we looking? How is this? Is this okay, or is this confusing? I imagine lots of you are confused because I also have messed this up a bit. Yeah, this is the same thing as chiasmata. Am I going to do the seminar in April? Yes, I am doing the seminar in April, and I will not mess things up. So yeah, yes, exactly, Helene. So essentially, autosomal linkage, you should get 3:1, but you don't because of this random sort of crossing over event going on here. So this allele here should be linked to this one. However, because they're swapping, you end up with this one here, this big B, taking the place and linking up with this big H. Okay, so that's what crossing over is all about. So in terms of looking at an exam question, then. So in fruit flies, the genes for body color and wing length are linked. Explain what this means. If you have two genes that are linked, what does this mean? Fadia, that is in totally forgot my word. It is in dihybrid crosses. Excellent, Lucy. Very, very good. So if the gene for body color and wing length are linked, it means they are both found. And then be careful with saying chromosome. You want to say same autosome. So they are both found on the same autosome. Okay, so autosome just means a non-sex chromosome. Okay, so a scientist investigated linkage between the genes for body color and wing length. He carried out crosses between fruit flies with grey bodies and long wings and fruit flies with black bodies and short wings. Figure 1 shows his crosses and the results. So G is dominant for grey and G is recessive for black. N, or big N, is dominant for long wings and little n is basically recessive for short wings. So I've got a cross where I've got a grey bodied long wing, so that one mixed with that one. And then the offspring are all that. And then it says these offspring were crossed with flies homozygous for black body and short wings. So if they're black-bodied and short-winged, it means that they are going to be little g, little n, little n. Okay, so it says here, so they've then done across here, and these are their results. So they've got 975 grey body long wings, 963 black body short wings, 186 of them, and 194 of them, right? So because they've said this is linked, it means that my chromosomes look like this. I've got a big G on one of them and a big N on the other. And on this one, I've got a little g and I've got a little n. So because they're linked, that's what I'd expect, right? So if we do the little cross let's draw this little cross out. So I've got little g, little n, and I've got big G, little g, and then big N, little n. In terms of what I'm crossing, it says use your knowledge of gene linkage to explain these results. So if I show you this cross, then it's going to be big G, big N, because they're linked, and little g, little n from this one. And I'm getting little g, little n from this one. I'm only going to draw it once because I don't need to do it twice because it's all the same, right? So I should be getting big G, big N, little n, and little g, little n, little n, right? So little g, little n is going to be my black body and short wings, that one. And then my big G, big N, little n is going to be grey body and long wings. Okay, so 975 of them, 963 of them. However, I've got these ones. So use your knowledge of gene linkage to explain these results. What do we guys think? So good. Actually, Lauren, you're getting the first mark really nicely here. So if you just say big G and big N and little g and little n are linked together, you will get a mark. I don't know why I'm saying you will get a mark. So they are linked. That is the first mark. So crossing over has created grey and short as well as black and long grey, and long found on the same chromosome, black found on the same. So Lucy, you are almost there, but we need to talk about it slightly differently. Lauren, you have also smashed this again. So parent GN, big G, big N, little n, and little g, little n, produced more GN, big G, big N, and little g, little n gametes. Perfect. Second mark. So most gametes are big G, big N, and little g, little n. So that is why I'm getting so many 9-7. That's why I'm getting so many of these ones and so many of these ones, because most gametes are them. However, what else do we need to say? Little g and big G, little n. Yeah, that isn't linked to that one, right? They're not linked. So how come we're getting them? How are we getting them then? Good. So crossing over is producing what? Due to crossing over, recombinant allele combinations occur, causing what, Blair? New combinations. What are my new combinations, though? Good, Lucy, you're spot on. Crossing over leads to, and that's actually a mark for saying that crossing over has happened. Big G, little n, and little g, big N, and we can say forming, let's say gametes. Forming the grey body, short wings, and black body, that's do it the other way around, long wing, black body. Lovely. Okay, so if these genes were not linked, what ratio of phenotypes would the scientists have expected to obtain? So if they were not linked, what would I expect? So I've seen someone do this. Lovely. Someone has told me. So as opposed to this Punnett square, if they weren't linked and I had big G, big N, little n, then these are all my possible gametes. So big G, big N, I'd have this. I'm going to struggle to fit this in, aren't I? Where can I put it? I'm just going to put it over here and you guys are going to have to deal with it. So big G, big N, big G, little n, which I haven't got yet. Big G, little n. So big G, big N, big G, little n, little g, big N. Don't know that yet. And little g, little n. I do have that. So in terms of my combinations, I've got big G, big N, little n. Little g, little n, little n. I've got big G, little n, little n. and I've got little g, big N, big N. So grey and long, that one. So that's one grey and long. That one is going to be black and short. This one is going to be grey and black. And this one's going to be black and long, right? So it's 1 to 1 to 1 to 1. So I'm going to rub all of that out. So I've actually got space for my last question. But you guys are totally right in saying that this one will be 1 to 1 to 1 to 1. So well done. And then finally then, which statistical test would the scientist use to determine whether his observed results were significantly different from the expected results? My genetic crosses look weird. That one was messy, to be fair. It wasn't that weird. Lovely, lovely, lovely. Guys, you know this is good. So if you are doing any of these genetic-y questions, if they ask you for the statistical test, you want to say chi-squared, it always is. And the reason for that is because you are comparing observed with expected results. So you have two categories and you are comparing them. So categorical data, some of you were saying. So this is because you are comparing observed and expected results. And I'm going to write in brackets categories. Right, and you're finding, you're basically seeing if there is a significant difference between the two. Okay, cool, right. I was going to go on to epistasis. However, looking at the time, I'm actually not. So I will go over epistasis probably with my next YouTube session. I might do like, actually, no, I'm not, because I've already made my next YouTube session. I might do another one in the future of like epistasis and Hardy-Weinberg, because they sort of go together. So let's do that. I reckon that'd be a good shout. And I'll do a stats thing as well. I'll do a stats on the view as well. That could be a good one. Epistasis and stats. Let's do that. How do you get the question? Sorry, but that's in the question. How do you get the marks? In this one, yeah, fine. It does talk about the observed and expected. Does it say expected at any point? It does. Yeah. So you want to say that you are basically comparing the categories. So you're using this because you're comparing these two categories. You're right that it is in the question, so you wouldn't get that. So you're talking about categories. But yes, awesome. My second bullet point, comparing observed and expected results. That's just it, really. And yes, synoptic essays, I can do them. They're really, really straightforward, Helene. So what I'm going to do then, guys, if I keep all that there, I am going to try and sell you this bad boy. There I am. Look how happy I look. Oh, I can't draw on my face. That's really sad. Anyway, so for those of you that don't know, and I'm sure loads of you do know this because we are advertising it like crazy at the moment, we are doing some like exam preparation seminars in Imperial College in London. So for any of you who are interested, basically me and the other Snap Revise people who I'm sure you've seen videos of are doing these sort of crash courses where I'm going to go through basically as much A-level biology as I can in April so I can get you ready for your exams. So I'll be going over things like exam technique, protein, cell division, mass transport. I'll be doing a year 13 one as well. Let's have a look. What am I doing for that? I'm doing photosynthesis, respiration, inheritance. I'll be doing all of this. And I'll make sure I've got that slide looking right so I can go over it without being confused. DNA technology, so all the PCR stuff, all the fingerprints, DNA profiles, all that. Ecosystems, harder calculations, that's all the stats in that one as well. And yeah. So we'll be doing that in the near future. So just so you know, the tickets are quite expensive. I totally agree before anyone tells me that. They are £99. But if any of you want to go to more than one of these, I think it's like you will get some discount if you basically email. So all of them, look at that. Look, there's some chemistry ones, 2 chemistry, 2 maths, 2 physics. And it's all going to be in lovely, lovely Imperial College, which I'm sure if you guys have never gone to a lecture to, it could be quite interesting, actually, because this is probably going to be your life for the next few years. So, what are we saying? Aisha, I swear I answered that. You live too far. For those of you that do live too far, I think there is, I think we might be doing this thing where we might try and, in fact, actually, I'm going to say stum about that for now. Ignore me. And yes, let's all pray that coronavirus doesn't get us first. Aisha, you can keep repeating it, but I don't, I'm not, hang on, bear with me, Aisha. Yeah, so I know it's a lot of money. I think the thing to think about for this is pretty much all of your education after you leave school is going to cost a ridiculous amount of money. So if you're even considering going to university, I was told when I went that every single lecture you ever go to will be £50 per lecture. right? And that's an hour. An hour of your time will cost £50. And that's if you do a degree like me. So I did biology, where you have loads of lectures. If you do like English and you're paying nine and a half grand a year for, what is it, like 4 hours of content, 4 hours of contact time a week, you're paying a lot more than that. So I know it's expensive, but education just happens to be expensive, I'm afraid. And I live in a lovely flat, and I would very much love to keep this roof over my head. So needs to be. What else are you saying? Oh, is it happening in another location? I don't think it is this year. I think next year we're going to try and do one up north a bit. But yes, I agree. We are really interested in doing it. And I don't know about being streamed online at the moment. So my second point, Aisha, I did say this like twice. You're doing chi-squared, and you are comparing. Oh, you're talking about this one, aren't you? So big G little n and little g big N are gametes which are formed. Basically, because of crossing over, this leads to these gametes being formed. That's what I'm saying. Other physics web classes too. I don't actually know if I know that. But anyway, guys, I'm going to call it a day there, and I will catch you at some point in the near future. Why is diarrhea a genetic condition because family genes? I've never heard that in my life. Second bullet point. Oh, most gametes are big G, big N. Yeah, because they're linked, most of the gametes are either going to be that or they're going to be that. That's what I'm saying. Anyway, no worries, guys. I hope you all have a lovely evening, and I will catch you at some point next week. I'll catch you on Wednesday next week, yeah? So next time I will be going over... Oh, I literally made it yesterday. I should definitely remember this. What was I going over? Oh, I'm going over stuff to do with the heart. So I'm going over heart stuff and hemoglobin. People don't like hemoglobin, so I thought I'd go over that. But the lesson after this will be epistasis and the statistical tests. So I'll go over those. Awesome. Right. Have a lovely evening, guys, and I will see you next week.